Hi, this is a video about the physiology of blood. The primary function of blood is to deliver oxygen and energy to tissues and remove carbon dioxide and waste products. It is also important for the defense and the immune systems, as well as regulation of temperature and the transport of hormones and signaling molecules between tissues. Now, blood consists of plasma and blood cells. Red blood cells contain hemoglobin and transport respiratory gases, whereas white blood cells form part of the defense system. In adults, all blood cells are produced in the red bone marrow. Platelets are also called thrombocytes. They are a component of blood whose function, along with coagulation factors, is to stop bleeding by clumping and clotting blood vessel injuries. Platelets have no cell nucleus. They are fragments of cytoplasm that are derived from megakaryocytes of the bone marrow. Plasma contains several important proteins with a total concentration of 65 to 83 grams per liter. Most of these proteins are synthesized in the liver, with the exception of gamma globulins. At a pH of 7.4, they are mostly in the acidic form. Their ability to accept or donate hydrogen ions means that they can act as buffers. Plasma proteins have important transport functions as they bind many hormones, example cortisol and thyroxine, as well as metals like iron. They are classified into albumin, globulin, and fibrinogen fractions. Globulins are further classified as alpha globulins, beta globulins, and gamma globulins. Red blood cells, also known as erythrocytes, are biconcave discs approximately 8 micrometer wide and uniquely have no nucleus. They therefore cannot repair themselves and have a lifespan of only 100 to 120 days. The shape and flexibility of red cells allow them to deform easily and pass through capillaries. Importantly, they contain hemoglobin, which is responsible for carriage of oxygen and also plays a role in acid-base buffering. Red blood cells are formed by a process called erythropoiesis. They originate from committed stem cells in the bone marrow of adults. In the fetus, they originate from the liver and the spleen. Erythropoietin, abbreviated as EPO or EPO, increases the number of committed stem cells and promotes production of red blood cells. Erythropoietin is produced mainly by the kidneys in adults. In the fetus, it is produced by the liver. The key stimulus for increased erythropoietin is low oxygen, also known as hypoxia. Stem cells differentiate into erythroblasts. These are the early normoblasts. They are relatively large and they are approximately 15 micrometer and they have nucleus. As differentiation proceeds, the cells shrink and hemoglobin is synthesized, which requires iron, folate, and vitamin B12. In the late normoblasts, the nucleus breaks up and disappears. The young red cells shows a reticulum on the staining, and therefore we call it a reticulocyte. As it ages, the reticulum disappears and the characteristic biconcave shape develops. Normally, 1 to 2% of circulating red cells are reticulocytes. This increases when erythropoiesis is enhanced, example by hypoxia. About 200 billion red blood cells are produced from the marrow each day. The spleen holds a reserve of red cells that can be released following blood loss. Red cells are destroyed by macrophages in the liver and spleen after 120 days. The heme group is split from hemoglobin and converted to biliverdin and subsequently to bilirubin. The ion is conserved and recycled via transferrin. Now, transferrin is an ion transport protein. The ion can also be stored in ferritin. Bilirubin is a brown-yellow compound which is excreted in the bile. An increased rate of hemoglobin breakdown results in excess bilirubin, which stains tissues such as the skin and the sclera of the eye, commonly known as jaundice. An inadequate amount of red cells and hemoglobin is called anemia. This is commonly a result of hemorrhage, example in heavy menstruation, but also occurs when the diet contains insufficient iron, folate, or vitamin B12, or they are poorly absorbed in the gut. Anemia is also caused by abnormalities of hemoglobin, indicating thalassemia, which is a sickle cell mutation, as well as leukemia, which is a cancer of the white blood cells, or the leukocytes, in which blood cells multiply rapidly and inadequate room is left for red blood cells to develop. Red blood cells have surface antigens that can react with specific antibodies in the plasma. The antigens and the antibodies are determined genetically. This forms the basis of blood groupings or blood groups. The most important systems are the ABO system and the resource system. Matching of blood groups is essential during blood transfusions. This is because giving red blood cells with a different antigen to that of the recipient will cause reaction with antibodies in the plasma, leading to agglutination and hemolysis. The resource system is important in pregnancy. This is because a resource-negative mother can be sensitized and therefore produce antibodies to the red cells from a resource-positive fetus during birth. This can be a problem for a second pregnancy with another resource-positive fetus where the antibodies cross the placenta and cause hemolysis of the fetal red blood cells. This can lead to severe complications. White blood cells, also known as leukocytes, defend the body against infection by foreign material. The white cell count increases greatly in the case of an infection or disease. Three main types of leukocytes are present in the blood. These are the granulocytes, the lymphocytes, and the monocytes. Granulocytes are further classified as neutrophils, which have neutral staining granules, and eosinophils, which have acid staining granules, as well as basophils, which have a basic staining granules. All these leukocytes contribute to inflammation by releasing mediators. Neutrophils have a key role in the innate immune system. They migrate to areas of infection by chemotaxis within minutes in order to attack and destroy bacteria by phagocytosis. They are a major component of pass from inflammation. Neutrophils live for approximately 6 hours in the blood. They actually live longer than that in the tissues. Eosinophils are less motile but have a relatively longer lifespan, and they phagocytose larger parasites. They are increased in allergic diseases, to which they contribute by releasing inflammatory mediators. Basophils release histamine and heparin as a part of inflammatory response. They are similar to tissue mast cells. Lymphocytes originate in the bone marrow but mature in lymph nodes, thymus, and the spleen before returning into circulation. Most lymphocytes remain in the lymphatic system. Lymphocytes are critical components of the immune system and are of three main forms. These are the B cells or the B lymphocytes, which produce gamma globulins, the T cells, which coordinate the immune response, and the natural killer cells, which kill infected or cancerous cells. Monocytes are also phagocytes, but they have longer lifespan than granulocytes. After formation in the bone marrow, they circulate in the blood for about 72 hours before entering tissue to become macrophages. Unlike granulocytes, they can dispose of dead cell debris. Macrophages form the reticuloendothelial system in the liver, the spleen, and the lymph nodes. Thanks for watching. Please like this video and subscribe to the channel. More videos coming up. See you in the next one.